Hello, everybody. Welcome. I'm so glad to have you here today. We are going to give folks a couple minutes to just get logged in and all set up. If you wouldn't mind putting in the chat where you're calling in from today. And just please make sure you mute yourself. So we have a lot of students on today, which is wonderful, but it will get very noisy if people are not muted. All right, getting those dings. Awesome. Welcome, welcome.
There's no volume.
Or we have several states already. Yeah. And schools, feel free to use your camera if you want us to see your class or not. That's totally up to you. But we are asking you guys to just stay muted during the presentation. And then actually, Sue, that made me unmute you.
And why we don't have any volume here?
All right, let's see. I think we're getting a couple people still logging in. Sorry about that. Just trying to manage the chat. All right, welcome everybody. It's so great to see so many people here today. So I know we have some people who are coming on their own just to hear the wonderful talk, but we also have a lot of classrooms joining in, which is wonderful. So we especially want to hear questions from our students who are working, who are listening today. So those questions, you guys can either share them in the chat as we go. Maybe your teacher could type it in for you, or you could type it in, or you can save them towards the end, and hopefully we can have some time to hear from you and hear your voices as well. So this event, like many of our events, is sponsored by the USA Blue Schools. We try to have about 4 webinars a year with scientists and other marine professionals, and to allow them to connect with students, and so that students can hear some of the things people are working on that are related to the ocean and marine science and other ocean exploration and other areas as well. So if your school is not already a blue school, I just want to tell you real quick what a blue school is. So blue schools are schools that are committed to improving ocean literacy and taking on action projects. And so some of our schools have done whole displays teaching others about marine debris. Other schools have mapped their watershed. Other schools have done art galleries about ocean literacy. So many possibilities. So if you are interested in becoming a blue school, you can register on our site, and I'll put that registration form in the chat a little bit later, and then we'll welcome you to the network and share a little bit more. So Dr. Kalina, I see that you're in New Jersey with your 5th grade. You missed some of it, but we have people from Connecticut, Rhode Island, New Jersey, New York, California, Maryland. So we have a good group here today. So, and from all over, which is wonderful. So I'm gonna stop sharing, and I am gonna turn it over to our scientist, Dr. Smith, who's here with us today. Dr. Smith is at the Mystic Aquarium, which is in Connecticut, and I know we do have some Connecticut schools here today. But for my friends who are in I see a bunch of New Rochelle people, way to represent New Row. I like it. For my New York and New Rochelle people, you're actually not too far. It's not your closest aquarium in Connecticut, but it is on the eastern side of Long Island Sound. You're on the western side of Long Island Sound. And okay, Dr. Smith, I'm going to turn it over to you, and I'll let you tell everyone a little bit about your career and then about our topic for today.
Okay. Oh my gosh, this is incredible. I've never had this many participants on a webinar.
Yeah,
Um,
we have a big group today. This is great. We're so glad to have you on.
would you be able to mute the doorbell for me?
I will attempt to.
Okay, got it.
Just
Well, I might
distracting.
actually be able to do that too.
Okay. I might be able to do that in the second.
Oop, nope.
I don't know if that's going to hit the doorbell or not, but
Yeah, okay.
I will try.
All right, cool. Yay, this is so exciting. Thank you so much for having me. Can you see my screen?
Not yet.
Oop, okay. I swear I've only done this like 2,000 times. All right, how about now? We're good? Awesome. First of all, thank you so much for having me. I am amazed at the amount of participants we have. It's so awesome. My name is Sue. I'm a scientist, one of 5 scientists here at Mystic Aquarium. And I'm going to tell you a little bit about some of the work I do as a scientist at the aquarium. And a lot of cool data, but a lot of gross samples. But I hope by the end of this, I can sell all of you in terms of the power of poop and other gross stuff in the sciences. So just to take a step back, because we have so many folks from everywhere, which is so awesome. We're in Connecticut, so we're continental US in the up northeast corner. And Mystic Aquarium is all the way kind of at the mouth of the Atlantic Ocean with this beautiful body of water, Long Island Sound. And I've actually been going to Mystic Aquarium since I was a nerdy 9-year-older, 10-year-older. I've always loved the ocean. My favorite first toy was a microscope. I loved going to Long Island Sound, learning about the snails flipping over rocks, looking at all the crabs underneath, all that good kind of gross stuff. And I love school, but after around high school, my grades kind of went off a little bit, and I basically didn't think that I had what it took to become a scientist, which was really sad because it's something I'd always wanted to do my entire life. And so I had a number of jobs, worked at guitar stores. I ran a bowling alley for a while. I sold computers. I worked at a golf course. And then I realized, no, I can at least try to do this. And so after a few years of trying to find myself, went to Houstonic Community College in Bridgeport. I'm a big fan, big supporter of community colleges. It's a great way to get into college if you don't know exactly what you want to do just yet. And from there, I realized that I can absolutely do this. I'm not bad at math. I actually love math. I love physics. I love biology. I love sciences. And so from there, I was able to transfer into the University of Connecticut's School of Marine Sciences. And there I did everything from my bachelor's, my master's, my PhD. I stayed on as a postdoc. And I now I get to teach there every year as a professor. So, and my focus at that time was on these little microbes called ciliates, basically plankton. But now I get to still study the very, very small organisms, microbes, but I get to do that to learn about the health and biology of the very, very big animals like whales. But I always like to take this aside in this intro when I talk to participants from this age group, because as I found out throughout my career that the more scientists I meet, the more I realize how almost every single one of them has a very winding path to their career, right? So any of the limitations you think you might have right now to prevent you from getting to where you want to be, those can all be overcome. It doesn't matter when or, or where you are in your journey, you can always get there. So at the aquarium, I have a lot of different studies, and I am lucky enough to study a lot of different animals. But my primary studies involve the beluga whale, the African penguin, which we'll talk about today. But I also had the opportunity to study sea turtles and seals through our animal rescue program. So if any of you have been to most aquariums, anything that's basically on the coast, you may not know that they often have rescue programs, meaning they will find locally stranded animals, stranded sea turtles, seals in our area, at least, and take those animals in. And while they're going through rehabilitation, I'm able to get samples from them. Our whole team, our research team, is able to get samples from them before they're released back out into the wild once our veterinarians clear them for release. And that's a really cool opportunity that you really don't get anywhere else, to study these wild animals. And then I also am lucky enough to have a lot of different collaborators throughout the world to get some really cool samples. Sometimes, I'll get here on a Monday and I'll have an email that says, someone just delivered 400 walrus poop samples with your name on them. And they want you to, study the stress hormone, corticosterone in them. So that's also a really fun part of my job, is I get to collaborate with so many other different, not only scientists, but also veterinarians, biologists throughout the world. And we can study these animals, and what I'm focused on studying these animals and their health and biology is through looking at, number one, their microbiome. If you haven't heard of the microbiome, it's basically all of the microorganisms that live on or in an organism. And in humans, we've learned a lot in just the past few years that you can tell a lot about the health of an animal, like human, by studying their microbiome. And in that, and by using other tools, I also look at disease, stress, reproductive health, determining sex in an animal if it's unknown. I also look at the diet of animals. That's really important in wild animals, with climate change and overfishing being such a major stressor on these animals. Knowing what they're eating and their relative diet is really super, super important. And all of that kind of comes together to pair with data that we have from veterinary specialists to kind of paint this picture of overall health. Now, the gold standard sample for studying health of an animal is blood. Just like human animal, right? If you were really, really sick and a doctor couldn't figure out what was wrong with you, the best thing that they could ask for you is to give a blood sample, because blood has a whole lot of secrets about the health of an animal locked inside of it. And so the positives of a blood sample is that basically with, a few drops, you can tell all about hormones, disease, health. It's very accurate, it's very reliable, and there's lots and lots of information. We have a history of information over time. The negatives is mostly that it's really, really hard to get a blood sample when you're not working with a human, but instead an animal that spends 100% of their time maybe in, the Arctic Ocean, right? Marine animals and marine science is kind of a lot newer than terrestrial animal science, just because they're inherently more difficult to access, right? You got to get on a boat, you got to assume you can find them. The average depth of the ocean is miles deep. So just getting out to them is already difficult. But then also, to get a blood sample, you need to capture this animal and draw the blood. And again, if we can get that sample, it can be super useful. We can learn a lot about the health and biology of the animal. If they're endangered, it might really allow us to get information where we can learn about them and help possibly conserve them for a longer period on our planets. But we don't really want to do that, right? We don't really want to capture this animal that's probably already stressed out and take their blood, even if it will help them in the long run. And personally, even I am very stressed out by getting a blood draw. Even having to choose this background screen for this slide kind of gave me a little anxiety. So I totally understand taking a blood sample can be really stressful. And so even though it gives so much information, right now, my specialty is to try to develop methods to kind of get all of that health and biology data out of a non-invasive sample. And so I know we've already teased this, but the biggest, best non-invasive sample, the reason there's a heart next to it, happens to be poop. Poop, as gross as it is and as bad as this lab can smell some days, contains so much information about the health and biology of an animal. All those things I was talking about before, the microbiome, disease, diet, we can all answer those questions from the littlest little poop sample. But some other non-invasive sample types that we can also gain information from are respiratory exhalate, also called breath or mucus or snot. We'll get into that in just a minute. But also skin. Even in humans, every time you kind of shed, even kind of brush against your arm, you're actually shedding cells. In those cells, there's DNA, and DNA kind of has a lot of the secrets and information that would be really useful to biologists that don't know a lot about these animals. For our feathered friends, so especially for my work with the penguins, feathers, even molted feathers, so the feathers that fall off the bird throughout their natural molting cycle, also can hide little secrets and pieces of information in that material. Same thing with the eggshell membrane. So after a chick hatches, there's always a little bit of membrane left in. Just like, you know, if you crack a chicken egg, there's kind of like the nasty little piece left over from the yolk. There's a lot of information in there about that animal's sex, even about their diet in some cases from their parents. And then another non-invasive sample type that can actually tell us about the health of animals is looking at their habitat. So again, this is of course not invasive. We can filter water and learn about all the microbes that this animal is constantly interacting with, and basically kind of do like a little forensics case on where this animal has been and what they're living in. And the beauty of this, and why I'm so passionate about this, is because this allows us to not only study the animals that we're in contact with, like at the aquarium, but also animals in the wild without possibly even having them know that we're there. So that's kind of the pie in the sky goal, is to learn everything we can about them without bothering them in any way. And so I've already hinted about this a little bit, but we can learn a lot from these non-invasive samples. So pathogens are basically, COVID-19, you could think of the virus, that's COVID-19, you could think of as a pathogen, bacterial pathogens, things that make you sick, cold, flu, viruses, persistent parasites. You can learn all about them through these non-invasive samples. Diet analysis, again, very, very important in a time of overfishing when we are drastically shifting what these animals rely on as their food. Dysbiosis might be a new word to you. Dysbiosis really refers to the microbiome and the bacterial assemblages or community of different species. So I don't know if you've ever taken an antibiotic, but if you take an antibiotic, sometimes your stomach gets kind of a little bit weird. And that's because something called dysbiosis occurs. It disturbs the biology, right? Bio meaning biology. And that can be okay if it rebounds after that. But sometimes in an animal, especially if they're really, really stressed, it doesn't rebound. And that can cause stress. It can cause a lot of immune system issues. We can also learn a lot about what treatment to give an animal. So at the aquarium, this is a really good example. Aquarium, a zoo, even if you take your dog or cat to the vet, if they have a bacterial infection, it's really, really difficult because of antibiotic resistance to know what kind of medicine to give them. What antibiotic do I give them? And we can kind of answer that question through these samples, through these data. And then microbiome, just studying again, those are all of the organisms, the microscopic organisms on or in certain areas of an animal. We found out in just the last under 10 years in humans and other kind of land animals that microbiome is linked to basically everything, linked to heart disease, some cancers, basically anything to do with your immune system. It also can affect the way that we age, the way that we think, the way that we interpret the environment. So microbiome is super, super important. And just in humans, we're just kind of learning about this now. And so for marine organisms, which again, inherently very difficult to access, we know basically nothing. So microbiome is something I'm really, really interested in for all of these different animals. Genetic sexing is basically trying to determine whether that animal is a female or male. looking at their genes or their DNA, right? DNA being that ATCG code that's inside all of us, inside all different organisms, from the littlest bacteria to, the blue whale. And inside that DNA is just all of the secrets of that animal's biology and also their health. In terms of transmission, something we're really interested in for wild animals, and particularly ones that are endangered, is how these, if you have a pathogen, for example, bacteria might be transmitted in the environment. During COVID-19, that was a really, really big science, right? Because the number one thing you kind of want to learn is how fast does this get from one organism to the other? And so that's something that we're trying to figure out with a lot of animals, like, for example, birds, avian influenza. If you heard of bird flu, that's a really, really big problem. And so if we can understand how that's transmitted, we can control and we'd be able to, lead to less culling of stocks, and possibly be able to stop that in its tracks. And again, it's just easier to sample wild populations using non-invasive samples. And it's really what we want to do. We don't want to, again, capture this animal that's likely already stressed and take that blood sample or take, a chunk of skin. We'd rather, scoop some poop or take a feather. And again, animal doesn't even know we're there, but we can help them. And so I'm going to talk a little bit about the beluga whale work, and then I'll talk a little bit about the penguin work. So the ways in which we do non-invasive sampling of our beluga whales is through swabbing. So if you've ever had the dreaded COVID-19 swab up the nose, that's basically what I use to sample a lot of these animals, except it's not as invasive. So swab, just like the ones you've probably seen. We take this, we swab their poop, we swab their skin, we swab kind of the outside of their blowhole, right? They don't have a true nose, they have a blowhole instead. The exhale, I'll show you a video exactly what I mean about that sample. We also swab their oral environment, their mouth. And then, like I said before, we also sample the habitat, in which case, and for whales and a lot of other cetacea, is the water. And it might seem like we're trying to get a lot of different parts on one animal, like we're over swabbing, but it turns out, especially in terms of microbiome, that you have, including humans, animals have totally different microbes that live in your mouth versus your skin versus your face versus your feet versus your gut. The gut microbiome we found is very, very important in everything that I kind of talked about earlier, the immune system. And so we're learning, basically for the first time in these animals, that it is very diverse and different, the assemblage of microbes, the different species that are located in all these different areas. And so, like I was talking about here, that exhale, that blow, this is really a big way in which we sample from our animals in the wild. But what I'm going to show you is an example of how we sample from our own animals at Mystic Aquarium. And this is Keela. She's my favorite. Don't tell Gina or Natasha. So she's, of course, trained with one of our excellent husbandry artists to exhale on command. So all she's doing is the equivalent to you kind of exhaling out your nose onto a petri dish. She always goes, yeah, after. So all she's doing is exhaling onto a petri dish, a sterile piece of plastic, basically. And then I use that same swab that I used for the rest of the samples to collect that sample. And then I take all these swabs and I send them for what's called DNA sequencing. If you've never heard of that, it's basically taking all the DNA, all the information that's trapped in not only information DNA from the host, the cells from the host, but also all of the bacteria, viruses, fungi, protists, and turning that into that ATCG code, that code of life DNA, where we can say exactly who's on that plate. And to give you an example of, even though that plate might look clean or just like it's got a little layer of water on it after Keela gives her sample, if we take that plate and put it under the microscope, turns out there's lots of life in that plate. And this isn't even bacteria. These are actually, they're microbes, but they're eukaryotic microbes, right? So bacteria are prokaryotic. humans, animals, other animals, grass, plants, they're all eukaryotes. So these are single-celled eukaryotes called ciliates. And so this is in one little mucus snot sample from one of our whales. We see hundreds of millions of these little tiny organisms called ciliates. And you can imagine if you were a vet and you saw this for the first time ever, you might be freaking out a little bit and thinking, oh gosh, this looks pretty nasty. And by the way, everything in the background here, that's all beluga snot. This might be a skin cell, which shows you how small these guys are, but everything else is beluga snot. And so at first, because we just know even now so very little about these animals, vets everywhere kind of thought this was a sign of a disease, right? If you go to your doctor and they found that in your mucus, in your lungs, or in your nose, they might be worried. But because at that time I was studying these microbes, I was easily able to tell through some electron scanning microscopy. So all that picture all the way to the right is an SEM or EM, electron microscopy. If you're a student or anybody who has never looked up electron microscopy, just Google that. There's the coolest pictures on Earth. And you can get really, really fine details, and you can learn a lot about the smallest forms of life or non-life, even viruses, using this imaging method, but also by staining them. So this is just one of those organisms, one of these single-celled ciliates. We're able to stain them. That round thing in the middle is their nucleus. We were able to very quickly say that these are not pathogenic parasites. The theory that they are feeding on the respiratory tract or the skin cells of these animals is impossible, because through our analyses and also through some DNA analyses looking at what is in their gut, the really, really small bacteria, we were able to say they're not even capable of eating the respiratory system of cetacea, of whales and dolphins. Instead, they're far more likely a cleanup crew. They're eating a very, very specific family of bacteria that loves to just live in big goopy mucus piles from our whales. And since then, we've found these in other animals, other whales throughout the world, essentially. So they're really kind of a cleanup crew, so to speak. And so I've talked a little bit about how I've shown you videos about how we do this with our own animals. And I've also been talking about the big goal is to use these methods to kind of learn about these animals in the wild, beluga whales, for example, in the wilds. Obviously, a beluga whale in a wild population is not going to come right up Aquila and put their beautiful melon, the top squishy part is called as a melon, and yes, it does feel like a marshmallow, up on the beach and exhale 3 times, after being trained for years. So we have to be a little bit more creative. So if you want to get our beautiful, lovely mucus sample in the wild, there's a few different ways people have found to do this. One is by using drones. So if you have just an everyday drone and you take these same dishes, these Petri dishes, and you glue them to the bottom of that drone, you wait out on a boat and you go and find a pod of these animals. You can fly that drone over that pod, and when they come up to exhale, we can catch that sample, fly it back to the boat, and I can get all that same information that I can get from Keela. And again, think about how beautiful this is, that this animal doesn't even possibly know that we exist, that we're there, we're not disturbing them, as opposed to, capturing them, taking their blood. Again, super useful information, but if we can learn about them, learn that they have a respiratory pathogen, we can learn perhaps, for example, that they have COVID, that would be really, really useful. And again, the beauty, we're not disturbing this animal. And this is crazy. I actually had to look this up after I came here and people told me that this is something that they do. This is mostly for larger whales, but for feces collection, fecal collection in the wild, collection of poop for whales, a lot of people immediately ask, like, how would you do this in the wild? The ocean is enormous and these whales are only so big. How do you find that poop? Believe it or not, there are dogs that are trained to specifically scent feces of whales, of specific species of whales. You can actually put this on a funding grant if anybody is interested in that. And we haven't done this work for belugas. It's actually a little bit harder because belugas are such a small whale. They're one of the smallest whales on Earth. So normally for us to get these samples, we go up every year to Alaska and we find pods of belugas, and they're very interested in humans. So you just kind of have to sit still in a boat and you will get, 20, 30 of these animals come right up to the boat. And we have a big stick with one of these petri dishes and we just hold it over them. And I just got some results back from doing just that a few months ago. So that's kind of the power of having being able to have these animals at the aquarium or at zoos. They have to be there anyway. They have to live out the rest of their life there and they can't be released. There's no way they would survive out there on their own. They're validating these methods so that we can go on to learn more about their species in the wild and conserve them for longer periods of time on our Earth. Okay, so I'm going to talk a little bit about the African penguin work I've been doing. So if you're not familiar with penguins, there's, I think, 19 species of penguins, and most people always think penguins, they think cold, ice, Antarctica. A lot of those species of penguins are actually in warm weather. So the African penguin lives in temperatures 80 to 120 degrees Fahrenheit. They're in South Africa. And unfortunately, they are critically endangered. They were recently uplisted, meaning they went from endangered to critically endangered about a year ago today. And there's about 50,000 birds. I probably have to update this. There's likely even less than that. Scientists are estimating they have probably around 10 years left on our planet before they are considered extinct. Really, really bad news. The only way in which we can conserve populations like this is by getting more information, right? You can't save a species if you don't know what's ailing them. And the way that we're trying to do this is to develop those non-invasive tools, because again, we don't want to go grab a South African penguin that's already maybe has malnutrition or avian influenza and take their blood. But instead, if you could go and gather some poop, some beautiful guano from this animal and learn that they do have avian influenza, or they aren't eating what they're supposed to be eating, they're eating this other non-nutritional prey, we could learn more about them, and then we could do something. We could tell legislation, oh, you need to stop overfishing in this area because now these penguins are eating this fish over here. And so we need to kind of corner off this area of fishing so that we can allow those fish stocks to rebound, so we can allow this colony to also rebound. And again, we're in a really unique opportunity at the aquarium, at Mystic Aquarium. We have a habitat of African penguins where we were able to validate that these methods even work. Because before scientists can go to South Africa or Alaska or anywhere else in the world, the middle of the ocean, and spend the time and money to, take some poop sample or take a breath sample, you have to first make sure that that works. And you can't do that without setting these animals in this unique controlled condition, which is exactly what we did. So we would go to Mystic Aquarium, go out into the African penguin habitats, and we would swab poop. And they actually have guano. I don't know if you're familiar with this. You might have heard of guano from bats. Guano is feces, but it's also not only feces, but also urine or pee. So it's a mix of poop and pee, just when you thought it couldn't get any grosser. But turns out from that one little, on the left is pictured of one little kind of collection of one of these guano or fecal samples from these penguins. Oh, by the way, sometimes I put this picture in. Sometimes if the sample's really big, we have to have other tools to collect them. So at a certain time, we realized that slushy straws are really good to collect feces in the wild. You can sterilize them really easily. They're super, super cheap. But I haven't looked at a slushie the same. So from that one single guano sample, really tiny little sample, basically the size of like a quarter, we found out that we can measure these 6 different hormones. And these are the hormones that you would measure in blood. Like if you went to the doctor and they didn't know it was wrong, they might measure these hormones to tell about reproductive health, to look at your thyroid, basically your metabolism, to see how stressed you are. You might have heard of cortisol in humans, corticosterone, kind of same thing. Turns out we have been able to measure all of these, validate that we can measure, and then measure all of these from that same sample. Also, same exact sample, we're able to take even less of it and also look at the microbiome. Remember, microbiome being the community of all of those microorganisms living in that animal. And from this in the wild, we'd obviously be able to tell all about any kind of pathogen, bacterial infection, fungal infection, viral infection they have. Also be able to learn about their diet from that sample. And then the third thing we're able to do, again, with this one little poop sample, precious, beautiful poop sample, is we developed a genetic sexing test. Now, again, genetic sexing, genetic meaning we're looking at the DNA, and then sexing meaning we're able to use that DNA, that code that's hidden. There's in that beautiful poop sample, there's just a few cells from that penguin, and we're able to kind of see inside of that penguin cell and determine whether they're female or male. And that hadn't been done before. It had only been done in the blood, using blood from penguins. And this is really cool because if we can do this in the wild, we can better aid in trying to figure out just how many females versus males there are, which is really important. I'll get to in a moment. All right, so I'm not going to go over all of these data, but I just wanted to step through these quickly just to show you how much we can get out of these samples. So this is that kind of point one, that hormones. So we're looking at 6 different hormones, and on that x-axis, this is over the course of over a year. So we have 30 birds that were in this experiment, and we looked at their poop samples, and all of these on the side, you can kind of see those 6 different hormones over their molting periods, their breeding periods. And we found out how females versus males changed over all of this time. And we were able to look at, if they changed, if they were over a certain age, if they were undergoing a treatment. And we saw all these crazy spikes during breeding, during molting. They were very, very stressed during certain periods of time. And this, again, is something that nobody really knew because they didn't have access to the animals. And again, you aren't going to go to South Africa and just try to validate, these methods using wild animals. Just too much variability. And so we were able to just right off the bat learn that not only can we measure this in these animals, but we found these patterns that were distinct and were the same for a lot of the same animals over time. All right, for that microbiome, I know this looks like a lot of data, but basically we're looking at microbiome data. So lots of different colors here. On the right, it's okay if you can't see it, super, super tiny. What you're seeing is a kind of legend, right? So see this little tiny blue square on the top? That blue square is associated with, it means that it's a certain bacterial species. And so if you have one of these bars here, you could kind of think of it as a pie chart. And so, this bar all the way to the right is 90% of that bacterial species, if that makes sense. It's okay if this is a little bit too much. The point here is that with this analysis, I showed this to, I presented these data to our animal vet team and our penguin husbandry team, all the trainers that take care of the penguins every single day, because I don't get to work with them every single day. I'm in a lab or behind a computer. And I showed them these data and I said, some of these animals, so I don't know if you could tell, but there's some of these little boxed out areas, penguin 7, penguin 12, penguin 9, penguin 5. Some of these animals are anomalously, or meaning very, very different in diversity from the rest of all of the birds. And I don't really know why, but they have high dysbiosis, right? Meaning their kind of microbiome might be like a little bit messed up, and it's kind of sustained over time. And they said, oh, well, let me look up what bird penguin 7 is, penguin 12, penguin 9. Turns out that every single one of these penguins that I happened to find blindly, doing this kind of blind analysis, was associated with low diversity, had either a problem with their molting, had problems because they were becoming very old. We had, I think, one of the oldest penguins, Frank Woods can correct me on this at a certain point. We had one of the oldest penguins in Aquaria at one time, or they were undergoing antibiotic treatment. And so this was so cool, again, to validate, no one had really done this work, that the methods that we're using can absolutely work to signal that there is a health issue. And then finally, genetic sexing. So in a lot of birds and a lot of animals, but not most actually, you might see sexual dimorphism, right? Every scientific word can be broken down into 2 parts generally, a prefix, suffix. Di means 2, morph kind of means what you look like, right? Your morphology. And so for a lot of birds, for example, like ducks, even a 5-year-old could say, that's definitely, a boy duck, that's a girl duck, right? Or those two look different. That's sexual dimorphism. We can very clearly see that there's two different kinds. We could tell female and males apart. Penguins, the best penguin biologists in the world, cannot tell a female and male African penguin apart. They're sexually monomorphic, meaning they only have like one type that they kind of look like for the rest of their life. And so again, normally, if you want to tell sex, you would take a blood test. But instead, we were able to use that same poop sample. And it's okay if you've never seen one of these before. It's a gel from basically the result of that DNA sequencing, or something called PCR. But basically, we were able to do something where, with just the littlest amount of that poop sample, we were able to visualize whether or not that animal was female or male. Basically, if they had two bands, because they had two different chromosomes, sexual chromosomes, just kind of like we have XX and XY, they were female, and if they had one, they were male. And so now, instead of having to go out into the wild, take this critically endangered, emaciated animal, draw their blood, wait to send it back for analysis, I can tell you within a day whether that animal is female or male. And we do know all of the sexes, of course, of our penguins at Mystic Aquarium, but the purpose of this was to validate these methods so that we know that they work, so we can then help the wild colonies that are critically endangered. Because if you go to a wild penguin colony and it turns out they are 1% female and 99% male, that's really bad, because genetic diversity is going to go way down. And diversity in nature, in all things, really, really, really good. And it turns out, after doing some other tests, I was not able to only sex them using their guano, their feces, but also those molted feathers, and even those that eggshell membrane. So how can we use, now that we know all of this, we know that it works, what are we going to do now? Well, next week, next Sunday, I'll be leaving to go to South Africa, to Cape Town, where there's a rescue facility and wild colonies surrounding that facility that I'll be able to basically constantly, every day, be swabbing and collecting poop and maybe some feathers and eggshells to answer all these questions. So I'm going to take guano samples from all of these birds, and as many as I possibly can over 2.5 weeks, and send those samples back home, where I can sequence them, analyze them, and say, this, you have this percentage of females versus males, you have this fungal bacterial in this colony, you have a bacterial infection in this colony, they're actually only eating 10% of, this species of fish when they were just really eating 90% of it. And the whole goal of this, as always, is to aid in that conservation monitoring, because we cannot conserve species on our planet unless we can monitor them and learn about them. And so I will kind of finish up there. Just big thanks to, if you haven't been to Mystic Aquarium and you're possibly able to, I'd love for you to meet these people. We have the best people on earth working there. Husbandry team, we have some of the best volunteers in the world. Frank Woods, one of my favorite on the call today. Veterinary team, some of the best on earth. And so I want to thank all of them, as well as obviously the animals who donate their beautiful but stinky samples so that we can preserve their species in the wild. And I want to thank you for listening and for being interested in very gross but very fun samples. Thank you.
Very cool. Thank you so much. This was so interesting. I definitely learned a lot, and I suspect our students did as well. And I definitely learned much more about why poop and snot are so important to science. So that's great. Can we do a few questions? I know some of our groups have to go, and that's totally fine. We are recording, so you can always follow up on the questions later if you can't stay on. But again, if you guys have questions that you want to put in the chat, you can do that. Or teachers, if you want to raise the hand, I want to pick on some of our classrooms first. So Lisa, I'm going to push you to the back for now. Sorry. If we have any of our classrooms, if we have a student who maybe wants to ask a question. Again, you can put that in the chat or you can raise your hand. All right, maybe we'll pick on Lisa while we're waiting on our kids. Go ahead, Lisa.
Okay, thank you so much, Dr. Smith. That was a really excellent talk that you gave us. I also learned quite a bit myself. So I'd like to be able to take this back to my organization, the New York State Marine Education Association. But my question is, and maybe the students who are out there in those two classes that I see, you might be interested in my question, which is, I saw the drone hovering over the beluga whale, and it was getting prepared to take a sample of the whale's snot, right?
Mm-hmm.
So when the whale breathes out, I assume that the whole drone gets covered in snot, as well as maybe an area where the sampling does get trapped. Is that true?
Yes, correct.
So, so when you actually pull the drone aboard or land it somewhere, I would assume that the drone is very gooey, covered in a lot of snot.
Yes, that's a great question. So someone actually thought about this, and now I wish I thought about it first. They've trademarked it, the snot bot. You can buy this. It is protected, it is snot sealed and protected. And actually a big problem with any kind of tool in the marine environment is salt water, right? Salt water is like the biggest corrosive element ever. So it's protected from that too. That's a great question though. And you know what? Even when like, if you saw that video of me sampling Gila, if you don't close your eyes, you know, you're gonna get snot
Oh,
all
right.
over your face. I've thought about testing the microbiome of us
And,
as we go on sampling.
thank you. And a quick question for the students out there. I saw two dogs aboard two vessels, and I didn't quite hear it. What's their role?
Yeah, so dogs, another remarkable way that they use their nose is they can actually scent whale feces in the wild.
Wow,
Yeah.
interesting. Thank you so much.
Yeah, thank you so much. Thank you for your interesting questions.
Thanks, Lisa. We have a few questions. Let's hear from Mr. Arango's class. Can we hear from one of our students?
All right, thank you. Can you hear me?
Yeah.
Yes.
Thank you so much. This was awesome. We have a question. Come up. Here. Why don't you come up so the microphone can hear you. So,
Hi, guys.
Hi.
Hi.
all right. So, all right. All right. So, go ahead. Ask your questions.
If a whale is sick and if a whale is sick and can it still breathe if it's sick?
Yeah, that's a good question. So have you ever been sick?
Yeah.
You have a cold? Yeah, so you can still breathe, right? Depends how sick the animals get. Unfortunately, we often only are able to find animals in the wild once they get so sick that they can't breathe anymore. But if they are sick and they're early and we could still catch it, then we could learn about what they have, if they have a cold or the flu. That's a great question.
Really good question. A question from one of our other classes, Mrs. Bullock's class, is how big is beluga poop? And do you collect small samples or a big amount of beluga poop?
This is a great question. So as you might guess, the bigger the animal, the bigger the poop. And it's big, but it's also in a water environment, right? So it's generally pretty liquidy, right? The animals aren't drinking salt water. They're drinking water that they get through their food, but they still have pretty liquidy poop. It is also, I should have mentioned, bright, beautiful green. Poop, beluga whale poop is green. Also, their breast milk is green too. But yeah, great question. And you can imagine beluga whale, pretty small whale compared to blue whale, right? Largest animal to have ever lived. That's a big poop. I got to get my hands on some blue whale poop. Good question.
Very good question. Another question from Ms. Quinn's class is how do you know that those penguins will be extinct in 10 years?
Ooh, really good question. So the answer to that is the power of data, right? So if we've been studying, someone's been watching these animals for the last, I don't know, 200 years maybe that we've been living with them. And we know that there's half as many every year, then we could start to calculate and make a graph, right? Well, it turns out people have been doing that with all different kinds of projections, or basically different ways of looking at that data. And that line is going down really, really fast. And if we kind of look at that line and see how fast it's gone down over every year, we can say what's going to happen next year, what's probably going to happen the year after that. But that doesn't necessarily mean that's going to happen. People like us are going to try to get in there and make some changes. So hopefully you'll all be able to get to meet African penguins sometime in your life.
Thank you. It looks like Miss Collins' class has a question.
Did you hear her?
We hear you, but we don't hear a student. They make
Say it really loud. Say it really loud.
How does the whale get sick?
How does a whale get sick?
Yeah.
Great question. So whales can get sick just kind of like all other mammals can get sick, just like us, right? We're mammals, so they can get sick from eating the wrong food, all right? Eat too much Halloween candy, you're gonna get sick. If you don't eat enough, you'll get really, really sick. You can also get sick by being around somebody else that's sick, right? That's why maybe you've worn a mask before, or you're supposed to, when you sneeze, make sure you don't get it everywhere. So we know that animals get sick from one another. But yeah, their diets, if there's other kind of stressors going on, so if there's a lot of stuff stressing them out, maybe someone's like humans are moving in on where they nurse and breed, they can also get really, really stressed out. And if you stay stressed for a really long time, you can also get sick like that. But that's a really good question. But most animals get sick just like we do.
Great question. I see another question in the chat is what has been the most exciting finding since starting your research?
Yeah, great question. So I think the most exciting work that I've done has had to do with finally getting that genetic sexing, being able to tell girls and boy penguins apart using their poop. That I'm really enthusiastic about and interested in, just because it opens up kind of a wealth of data, a big opportunity to help to try to learn more about these animals in a way we never have. Yeah.
Great. We have a few questions about the lifespan of belugas. Maybe you know that, and if they're considered endangered or worried about them going extinct.
Yeah, great question. So just like kind of with humans, there's different populations of one species of animals. So for beluga whales, there's actually like 5 or 6 different populations, meaning all the same species, but they live in different areas. You can kind of think about living in Connecticut and living in California, right? And some of those populations, so some of those groups of beluga whales are critically endangered, meaning that there are only 300 of them left. 300 might sound like a big number, but it's a very, very, very small number, right? There's billions of people on the planet. And so, yes, there are some groups or populations of beluga whales that are critically endangered, and they are unfortunately on the road to extinction. But we are also actively studying them every single year and trying to figure out why. And I am just one person out of this team. Everybody else is also learning about the health and biology of these animals in other ways to help aid in their conservation, to try to answer these questions that you guys are asking, and trying to answer why they're doing so poorly. But overall, belugas are on the up and up. We used to hunt belugas, and some people still do. For subsistence hunting, they need to hunt, but they're kind of coming back from overhunting in the past. But yes, there are some critically endangered populations or groups of beluga whales.
That's helpful. Thank you. Okay, I think we have time for one more question. I see from Mateo and Mr. Arango's class, how long can belugas stay underwater?
Ooh, this is a great question. So it's I think maybe much more than you'd think, 30 minutes. I know that we do dive work with them. We have someone that just studies dive specialists of the world for this animal. And I know our animals can go to depth for quite a while, but that might be a question that you guys can answer for me.
All right, we'll have to take a look for that. Thank you so much. This has been so wonderful. I appreciate also getting all these questions answered. We had some really wonderful questions from our students today. So we're so glad you were all able to join us. Can we give one more big thank you to Dr. Smith for all of her knowledge and sharing it with us? I see lots of claps and virtual ones
Thank
and
you.
real life ones. Thank you so much. This was super fun. Thank you all for coming. And yes, safe travels. Have a great time in South Africa. I can't wait to hear what you learn working there.
Thank
And
you so much. And thanks to all of you guys. Remember, we need more scientists. So if you really, really like science, keep up with it, okay?
yes, and science is for everyone. So I
Yeah.
want to reiterate what you said earlier. So that is terrific. Everybody can be a scientist. So it's not always a straight path. So you can all do that if you're interested.
Yep.
All right. Thanks so much, everybody. Have a great rest of your day.
Thank you.
Bye.